All right. Sorry about that quick, short delay, but we're right on time still. So you're going to be talking into this microphone here. We're very pleased to introduce our keynote speaker, Dr. Joanna Fowler from the Brookhaven National Laboratory. Dr. Fowler is going to be talking on imaging brain chemistry and diseases of addiction. Thank you.
Hello, everyone. Thank you so much for inviting me. This is quite an honor, and congratulations on having this center. I think there's not many things that are more important than working on addiction and trying to understand the brain changes that occur when people get addicted to drugs, because it goes all the way from the molecules that are involved to the whole neighborhood, it's the whole social context, and I think we're realizing how complex it really is. So thank you for inviting me. Thank you to the organizers. And also, I have to tell you, I have nothing to disclose. So with that, I'm an organic chemist, so there's some chemistry spattered in here. Hopefully it's not too painful to see, but I want to explain a little bit about how we get images. So how do I change the slide? Oh, can I change it here?
Yeah, let me know. Sorry. Now just look at that.
Okay, let's see. Okay. All right. So addiction is really a very complex disorder. We have not only the effect of the drug on the brain. Is this pointer working? I guess it's not only.
A little bit.
Well, okay. I'll do it with hand waving. We have not only the effect of drugs on the brain, but we also have biology and genetics. We've also always heard of families of alcoholics.
If you can be over here, is that okay to see you?
Okay.
You can.
All right. I'm fine. I'll try not to wander around too much. So there's not only the effect of the drug on the brain, but there are also biology and genetics. Addictive substance abuse disorders tends to run in families, though we realize there's also a very strong environmental component. You can also train an animal to self-administer drugs, but you can put them in you can do this much more easily if you put them in a stressful environment. So a lot of things impact on drug abuse, not only biology, genetics, the drugs, and the environment. So we're realizing what a complex disorder this really is. I'm sorry, am I doing something totally?
Sure you are, Dr.
So what we use is a technique called positron emission tomography. It's a medical imaging method. And we use very short-lived isotopes, carbon-11, which has a 20-minute half-life, and fluorine-18, which has a 110-minute half-life. These are radioactive isotopes, and they emit an energy, very high energetic photons that can be imaged from outside the body. So we have different types of PET scanners. You've probably heard of a PET scan. I'm going to tell you a little bit about how you get a PET scan. So there are human PET scanners shown in the top. There are now micro PET scanners that can be used to image animals, image small animals. And then at Brookhaven, we've developed what's called a rat cap PET scanner that sits on the head of a rat, or an awake behaving rat, so that you can study behavior and brain chemistry at the same time. Now, let me just go through this rapidly. I'm going to show you images of cocaine in the brain and how it moves in the brain. And I can show you images of these in a normal person. It is because we can make these PET images with administering very tiny chemical quantities. So you're not changing any of the you're not having a pharmacological effect. So that's what this slide is all about. It requires working very rapidly. So a positron is a positive electron, and basically what happens is that when a positron decays, you get very energetic radiation, and it can penetrate the body barrier. So if you inject one of these things, you can image where it is, and you can image the movement of a drug like cocaine. So the most well-known radiotracer is a molecule called fluorodeoxyglucose. It's a fluorine 18 labeled sugar. It can penetrate the blood-brain barrier and mimic glucose. And if you inject this, you can tell where brain chemistry is occurring or where brain metabolism is occurring. And so basically, a PET scan is you can either make or buy a radiotracer like SDG. You inject it. You get an image by surrounding the body with a ring of detectors. This makes, with computer algorithms, you can get an image. This is a whole body image, and it shows where glucose metabolism is occurring in the brain and in the body. And if you can look at this image here, you can see the person's brain is lit up here. Their heart is lit up because the heart sometimes uses sugar for energy. You can see muscles lit up, but you can see a very special group that you wouldn't normally see in a normal person. This is a person with lung metastases from ovarian cancer. And this occurs because tumors also burn sugar. So your brain uses sugar almost entirely as an energy source. Tumors also burn sugar. So that's why this is such a useful tracer for cancer. So when we talk about addiction, we have focused in our group mostly on dopamine. Dopamine is very important in addictive behavior because all drugs that humans abuse raise brain dopamine levels, and they raise it in the nucleus accumbens, which is called the reward center of the brain. And they do it even, we're talking about all kinds of different drugs, nicotine, marijuana, heroin, cocaine, alcohol. They all elevate brain dopamine in this reward center of the brain. And what's interesting is dopamine is also elevated in feelings of well-being in anticipation of natural reinforcers like highly palatable food. So if you look at a very oversimplified cartoon of the brain, and you slice the brain like this, the cells that produce dopamine are deep in the brain in the ventral tegmental area and substantia nigra. They project to the reward center. They also project to the frontal cortex. And these parts of the brain are very important. The reward center, of course, is what I just told you about. Frontal cortex is also very important. It's called the executive control center. It allows you to anticipate consequences of behavior. So in an animal, you can drop a probe into the reward center and look at the effects of different stimulations on dopamine. For example, if you show an animal food, you get a small blip of dopamine. It'll maybe go up like 50% here in the lower graph. If you show an animal or you allow an animal to lever press for amphetamines, the amount of dopamine will go up to 1,100%. So you can see a difference between 50% and 1,100%. It's a huge difference. And if you equate this to pleasure, you can see how the drug would win out over a natural reinforcer like food. So what we've done is we've developed radio tracers labeled with these short-lived isotopes to look at various parts of the dopamine synapse. So if you slice the brain across like this is the frontal cortex up at the top. The nucleus accumbens is the two spots in the center, and the cells that produce the dopamine are down here deep in the center of the brain. So when the cell fires, it releases dopamine. So this is the synapse. This is where the action occurs. When the cell fires, it releases dopamine. Dopamine binds to a receptor. Sorry, this is so weak. The receptor is in blue, and when the dopamine binds to the receptor, that transmits a signal. So there are parts of the cell that are very important. The receptors are very important. It's important to know.
Joanna, I would use this just to point on my computer here.
Okay.
So that people can see it and then
all right.
venture off all that.
Okay. Okay. So what we've done here.
Go back.
Yes, sure. Okay. So when the cell fires, it releases dopamine. Dopamine builds up in the synapse, stimulates. Sorry, I'm so awkward today. Dopamine builds up in the synapse. It stimulates the receptors, which are shown in blue, and you get a signal, and the signal can be pleasure or movement. And so what's important here is, I'm sorry.
These. This one?
Yes, yes.
This is also the back.
Okay. And so the red element here, so I want to do this without moving it.
Right.
I'm going to use up all my time figuring out
Don't
how
worry.
to do this.
You're okay.
Okay. So the red element actually is a part of the cell that picks dopamine back up and stores it. Okay? So we would like to image the blue element and the red element. And so what we've done is we've developed these tracers that can allow us to look at these different parts of the cell. So we can look at the red elements with a tracer. It's actually, we put a C11 on the cocaine molecule. We can look at the blue elements with a different tracer. And we can look at glucose metabolism with the tracer FDG on the bottom. And we can look at enzyme activity. And we can also look at nicotine binding sites. So you get what we're doing. We're making all of these different molecules so we can look at the addicted brain in different ways. So we started this program in addiction with...
Go back to this one here.
Okay.
Then you put that down and walk freely.
Okay. I'm a wanderer.
Yeah. Okay. Put that in your pocket.
Sure.
Turn this off.
So when Nora Volkoff came... I'm sorry.
Is that on?
Can you hear me now? Yes. Okay. So in 1988, Nora Volkoff joined our group, and she was quite interested in drug abuse. She's now director of National Institute on Drug Abuse, and she had been working in the emergency room at the University of Texas, and she was doing scans on cocaine abusers who had come in, and they had stroke-like symptoms, and their brains had big areas of no blood flow. And so she was very curious as to, what is so powerful about cocaine that people will keep taking it even if it's causing all of these problems, which is kind of the definition of addiction. It's the loss of control over behavior, even if it's causing tremendous problems. So she came to us, and the first thing that she wanted to do is actually look at where does cocaine go in the brain. If you could actually follow that molecule around, what would you learn? And so cocaine has a very interesting mechanism of action. It binds cocaine and also methylphenidate, which is also called Ritalin. Both of these bind to the dopamine transporter. So if you see here the purple, that's the cocaine molecule. It binds to the dopamine transporter so that when the cell fires, dopamine builds up in the synapse. It stimulates the receptor, and you get a large signal. And we think this is what caused the euphoria from cocaine. So what Nora said is, would it be possible to put one of these carbon 11s on the cocaine molecule so we can actually see where it goes in the brain? And so we could, and this is chemistry. We actually use a cyclotron. We make the carbon 11. We put it in the cocaine molecule. And you can put it in there and just have a tiny, tiny bit of the chemical mass of cocaine. But you have this signal that you can use to image it. And so these were the first images of cocaine in the human brain. And this was slicing the brain like you go from the top of the head down to the base of the skull. And this is a rainbow scale. So where it's red is actually where that molecule is in the brain. So we've sliced from the top up here down to the cerebellum. And you can see that the cocaine is very much localized in what's called the basal ganglia, or the reward center of the brain, which was, cocaine's kind of a dirty drug. It binds to a lot of things, but we were very pleased to see how exquisitely this localizes in the brain. And the most surprising thing we learned about it is how much gets to the brain. A huge amount. You inject this, you inject cocaine, you smoke it, whatever, a huge amount gets into the brain. Not all drugs get to the brain very heavily. Some of them get there slowly, some of them don't get there into the brain very well at all. Cocaine is really a master at getting into the brain. That molecule is designed to get in. And one of the most fascinating things was when we can sit on the one slice, we can actually, with a PET scanner, sit on the one slice of the brain that has the reward center and look at it through time. And so this is like a time lapse through the brain, through the reward center. This is a one plane of the brain through the reward center, and these numbers you see are minutes. So this is 1 minute, 3, 5, over to an hour. And we see that it peaks really rapidly. So that cocaine gets into the brain very heavily, peaks very rapidly, which is what people want for reinforcement, but it also exits the brain very rapidly. And so we were working with this initially. This was a sub-pharmacological amount. That is, we couldn't see a behavior or a high from it. So we went into the literature. This was about 25 years ago we did this, and looked at have people actually mapped the time course of the high from an intravenous dose of cocaine. And indeed, they had in a laboratory setting in cocaine abusers. So we plotted our uptake and egress from the brain versus the high. And the uptake and clearance almost perfectly parallel one another. So we think this explains, this rapid, heavy uptake really explains the highly addictive properties of cocaine and its self-administration in a binge-like pattern. So they take cocaine, they get very high, it's very pleasurable. In a half an hour, the high is gone, they take it again until either they're exhausted or the supply is gone. And so, and this wasn't a new discovery, but we keep learning it over and over, but how fast a drug gets into the brain plays a really key role in whether or not it's reinforcing. Smoking and intravenous injection are the most rapid ways of getting drugs into the brain. And they are also the most likely to result in addiction. And so you can really think of the smoking of cigarettes. Smoked nicotine is very reinforcing. It's not replaced very well by a patch or by gum because those deliver it more slowly. So we started to ask the question, what is the role of dopamine in drug addiction? And then we started to look at the dopamine receptors, that is these blue elements down there, and asking questions as to what happens in the addicted brain, what happens to dopamine receptors, because that's kind of where the action occurs when the dopamine hits the receptor. And we found that on average, cocaine abusers, and these were heavy cocaine abusers, long-term heavy cocaine abusers, have an average lower dopamine receptors than control subjects. And we always have to regress with age because you're losing dopamine receptors as you age, and so we regress with age. But on average, and we replicated this in a number of populations, the cocaine abusers have lower dopamine receptors than the controls, which are shown in pink. So we went on to look at other addictions. We looked at cocaine, and here we have the control populations on the left here and the addicted on the right. And on all the things that we've looked at, we looked at cocaine, methamphetamine, alcohol, and heroin abusers. All of them have lower dopamine receptors than a control population, than an age-matched control population. And so Jean-Jacques Wang, a physician in our group, said, well, in obesity, people show the same kind of behavior. They can't stop eating, even if it's causing problems. So he said, let's look at some very obese individuals. So he started to look at obese individuals and indeed found the same thing. The control comparison, lean subject BMI 23 versus a very obese individual BMI 50, the dopamine receptors are lower in proportion to how heavy the person is. So we think that the reward circuit, that we have low reward stimulation in addiction, and that people are probably taking drugs to increase their dopamine level. So if you look at it, I've drawn an exaggerated cartoon on your left where you have many receptors here, and fewer receptors. And so you figure the cell fires and it releases dopamine. If there's not many receptors to catch it, you're not going to have that signal transmission, that sense of well-being. So, okay, so we have lower dopamine receptors, they get a weaker dopamine system. How does that affect function? And so we have our tracer, the sugar tracer, fluorodeoxyglucose. In the same subjects, we could look at both dopamine receptors and brain function. And indeed, we found that just as we get lower dopamine receptors in the cocaine abusers, there is also lower metabolism in the orbital frontal cortex, which is the executive control center here. And so if you plot one against the other, you can see the lowest metabolism has the lowest dopamine D2 receptors. So at the same time that the dopamine receptors system is weaker, you also have this diminished control from the frontal cortex, which tells you the consequences of taking the drug. So we asked the question, if you could magically increase these dopamine receptors, could you change addictive behavior? And we don't know how to do that in humans. There's some evidence that diet and exercise are very, very good for the dopamine system, but Peter Thanos in our group had some alcohol drinking rats. So he had a behavior that he could look at. And so what he did is he put extra dopamine receptors into these rats, put it on a gene, put the gene into the reward center, and you get a 50% increase in dopamine receptors after about 4 days, and it wears off after about a week. And lo and behold, look at what happens to the alcohol intake. The alcohol intake almost completely stopped. As the receptors wore off, they started drinking again. You could put another vector in and repeat the cycle. So if there was something we could do to change and strengthen the dopamine system, that would be really, really something important and a breakthrough. Now, there's another study which wasn't done in our group, but it's one of my favorites, is a study on social rank and vulnerability to drug abuse. So this was done at Bowman Gray University in Mike Nader's group. And what they had was a group of singly housed rhesus monkeys that were hooked up so that they could lever press for cocaine. And so they could scan them ahead of time and look at their dopamine receptor level. And they could look at their drug use. So what they did is they put them all together in a group house. When you put them together in a group house, some of them become the dominant animals and some of them become the submissive animals. And so then they scanned them again after 3 months, and they found that, lo and behold, the dominant animals actually got more dopamine receptors. Their receptors level went up as a result, just as a result of their environment, of their socially changed environment. And their drug abuse went down. So if you look at the green circle, the drug abuse went down, or the drug use went down. The subordinates, the drug use went up. The subordinates, the dopamine receptor levels stay the same. So it shows that dopamine receptors can change within the time frame of normal social activities, which I find really pretty impressive, and I think certainly for treating drug abuse, it shows the importance of a social context relative to the effectiveness of drug abuse treatment. So we've also been quite interested in tobacco smoke and looking at it from a number of different perspectives, and we've used a number of different tracers, which I show here, the chemical structures on the side. Smoked nicotine, you can put a C11 on the nicotine molecule like this, and you can look at how fast it goes in and out of the brain. Smoked nicotine, it goes in and out of the brain very rapidly, similar to cocaine. Very, very rapid. Whereas if you put a nicotine patch or gum, you'd see a much more shallow curve. It would get in there very much more slowly. So nicotine also gets into the fetus brain. So we had access to pregnant macaque monkeys for a while, and an anesthesiologist who worked with us. And so we put the label on the nicotine molecule, injected into into the mother and looked at the fetal brain, and looked at whether the nicotine gets into the fetal brain. And indeed, it does. This is a third trimester, and these were actually difficult studies to do because we wanted everything to turn out well for the little mothers. And you can see the baby's brain. It's labeled here. It's probably better than me in my mouth here. But you can see it goes into the thalamus, the part of the brain that contains the nicotine receptors. So certainly good evidence that the nicotine gets into the fetal brain. So this was a really nice model for looking at how drugs penetrate into the brain. So we've also been asking, and a lot of people ask, to what degree does tobacco smoke actually occupy those nicotine receptors in the brain? And these were some studies done out here at UCLA by Arthur Brody. And if you can label these, like now we label dopamine receptors and then PET scans. Now we're going to label the nicotine receptors with a different tracer, which is this one shown in the center with the fluorine 18 on it. And you can see, if you look here at this row, no smoking, you have the receptors unoccupied, the tracer binds, nice bright scan. If you have a low nicotine cigarette, you still get quite a lot of occupancy. So that means the nicotine is binding and the tracer can't bind. If you have a really low one of these de-nicotinized cigarettes, you see that these are totally blocked. So you see, this is the no smoking. This is the high. This is the real cigarette. Is the blue one, and you can see that they're totally occupied, about 80–90% occupied. But they also did a really interesting study on secondhand smoke. They did these same studies and then had one of the volunteers sitting in a car with a person smoking for an hour. And you see even the secondhand smoke, you can see a real effect on these nicotine receptors. So it's not that, it's not just something in our imagination. They measure a real effect on nicotine receptors. So nicotine affects not only the nicotine receptor, but it also affects this enzyme aromatase. Aromatase is the enzyme that converts testosterone to estrogen. And it's very important. It hasn't been studied very much. It mediates sexual differentiation in the brain during development. So during development in the fetal stage, all brains are female. They're all female brains. So the way you get male behavior on a male fetus is that this enzyme takes testosterone from the testicles of the male fetus, converts it to estrogen, and it's estrogen that masculinizes the brain. So if you knock this out or if you block it, you can have a male fetus with female behavior. So it's very, very, very important. And these enzymes that inhibit this are used to treat breast cancer. So we put a C11 on one of these molecules to look at this enzyme in the human brain for the first time. And this was some difficult chemistry that occurred to put this on the right molecule. The way you do this is you make a small part of the molecule here, this methyl iodide we call it, and you attach it here. But we only wanted one. We wanted the methyl group attached here, not here or here. And the way chemistry goes is that it's attached to all three places, so we had to do a difficult separation. But long story short, we got enough of this to start studies, and we started studies in the baboon. And here we're slicing the brain like this. And so what you're seeing here is, you see these two spots, but they're not the nucleus accumbens this time. They're the amygdala. It's an emotion center in the brain. So this molecule binds very much. The aromatase is very heavily expressed in the amygdala, and we do various ways to prove that it's actually binding to aromatase. And so we were able to start these studies in humans, and this is something that was very interesting, because we usually start these studies in our group of monkeys, and then we proceed to humans. And we found that the amygdala in humans is, here we go again, is very low. The aromatase is very low, and it actually occurs, the enzyme occurs mostly in the thalamus and in some of the areas of the brain stem. At any rate, we were debating when we did these studies whether should we exclude smokers or not, because we find so many interesting things with smokers. And it turns out, we started looking at the literature, and it turns out that nicotine inhibits aromatase. So we did some studies in baboons giving nicotine intravenously and found that indeed at levels that a smoker would experience, levels of nicotine in the bloodstream, it actually does inhibit aromatase. So this was very curious, and we said, you know, we've got to look at smokers. And these studies were done like in 5 years or so ago when we first started studying smokers. It was really easy to get smokers like in the early 90s, you know, heavy smokers. I mean, the good news is it's harder to recruit smokers for your research studies, which is a good thing. But we did manage to get some for this study. And cigarette smokers, human cigarette smokers, also have lower brain aromatase than non-smokers. So what does this mean? Could this account for any of the things we know about smokers? And remember, this, if you inhibit aromatase, you decrease the production of estrogen. So we think that now nicotine inhibition of aromatase could account for some of the observations that female smokers go through early menopause. They go through menopause about 3 years earlier than non-smokers. So we think there's the hypoestrogenic effects of nicotine on the aromatase that also cause not only the early menopause, but also lower voice in female smokers. It's not just sort of scratchy throat, it's actually lower voice due to these hypoestrogenic effects. So we also were looking at other effects, things that are in tobacco smoke that aren't just nicotine, and there are about 4,000 chemical compounds in smoke, including inhibitors of monoamine oxidase. So monoamine oxidase is an enzyme which breaks down neurotransmitters like dopamine, which is involved in reward, or serotonin, which is involved in mood, and norepinephrine, which is involved in arousal. And MAO inhibitor drugs, you all may have heard of these. They're also used to treat depression and also used to treat Parkinson's disease, because in depression, you want to prevent the breakdown of these neurotransmitters. And in depression, you also want to elevate neurotransmitters. That's what your SSRIs do. They elevate serotonin. So we can image the monoamine oxidase with different tracers in the brain, and you can see these patterns. Monoamine oxidase occurs in 2 subtypes. The A subtype here, you can image with a compound called chlorgeline, and the B subtype you can image with a compound called deprenyl. And this is a very different pattern than you see with the dopamine receptors. You see high enzyme activity in the thalamus, but you're looking actually at enzyme activity. And we started to look at smokers. This was actually an accidental discovery, as we found that certain normal people, we were studying normal aging, and you lose monoamine oxidase. You lose your glial cells as you age. At any rate, we found that smokers have lower monoamine oxidase in their brains than non-smokers. So if you look at over on your left, the group of non-smokers, smokers, and then people purposely treated with an antidepressant drug. So we were curious about this, and we don't know if this impacts on the behavioral effects of cigarette smoking, but there are things we learned when we started reading about some of the epidemiology of smoking. And we know, and this has been repeated many times, is that smokers have a decreased risk of getting Parkinson's disease. And this has been replicated a lot of times. it doesn't nearly compensate for the morbidity and mortality associated with smoking. But people have been knocking this around. Is tobacco smoke neuroprotective? And if you look at the monoamine oxidase reaction, you have your neurotransmitters, and they are broken down, and they produce hydrogen peroxide. That's this molecule here, which is the source of free radicals and reactive oxygen species. So if you have the smoker with their MAO decreased over a lifetime, you decrease the amount of free radicals, and you protect the neurotransmitters. So this may have this slight protective effect in Parkinson's disease. But the more interesting thing is who is at higher risk for smoking and the comorbidity of smoking with mental illness. So 44% of all the cigarettes that are smoked in the United States are smoked by individuals with occurrent psychiatric disorders. And 75% of the patients in addiction and mental health programs also smoke cigarettes. In fact, all of the scans I showed you with the cocaine abusers and the alcoholics and the heroin abusers and the methamphetamine abusers, only a handful of those were non-smokers. You can't really study human addiction without having a confound of cigarette smoking. So, you know, I want to ask the question, what's going on with this? If you look at some of the numbers, in the normal population in this country, about 1 in 5 people still smoke cigarettes. This is and it's about an equal amount men and women, as is the lung cancer rate right now. If you look at different countries, the rate is very different. The numbers in parentheses here are women. So it's very much, it goes very much by region and by culture. But if you look at alcoholics, almost all alcoholics are also cigarette smokers. Other addictions, like I just said, we could never recruit. We couldn't do any studies if we had to exclude smokers from their population of addictions to other substances. In schizophrenia, about 85% of people with schizophrenia also smoke cigarettes. In depression, about 80% of people who are depressed also smoke cigarettes. And in fact, this is a psychiatrist will talk about how difficult smoking cessation is in people with major depressive disorder. And in fact, one of the non-nicotine treatments for smoking or smoking cessation treatment is a drug called Zyban, and it's a renaming of an antidepressant drug called Wellbutrin because there's a stigma against taking an antidepressant. So if you call it something different, there would be more compliance. So it's a realization that some of the people who are smoking may be using it for symptom control and self-medication. So I just want to end up here with just a plea to people who are working on addiction. I mean, there's a public prejudice against addiction as being a moral weakness and a lack of willpower. And I think we can tell from brain images and from a lot of the studies that are going on right now with all of the tools we have, that there are real changes in the brain and that it really matters. It impacts public health probably more than anything else that we can think of. And it also requires a really multifactorial approach to the study. It requires not only like the molecular events that I'm showing you, but just, you know, the social event, what happens in neighborhoods, we need a constellation of all of these tools brought to bear on this problem because it is incredibly important. And I think you don't usually think of heart disease and cancer and addiction at the same time, but 30% of the heart disease and cancer are from smoking cigarettes. It's from addiction. It's from a disease of the brain. And we have these huge investments in cancer, in lung cancer, and heart disease, whereas we're really looking at this. Same with HIV. Is a large fraction of HIV came from intravenous drug use, sharing needles, risky sexual behavior secondary to stimulant abuse. You look at accidents and violence. With alcohol on board, there's a very large fraction of accidents and violence that occur when people are drinking. And then obesity, again, this loss of control over eating behavior. So all of you who work in this field, you really inspire me. I really value what you do. I value all of you who are going to go in and work in this field and think about it and observe the people you're working with, because it's really important. And so I think, you know, you can take it all the way from the physical sciences, and you develop these new tools. You can do to make new instruments for imaging. You can do new chemistry, so you can have new molecules. You think of something else you want to look at, you can make a molecule. You can use these tools to get new knowledge, and then you can use these new tools to find out things that we don't know and things that we're still puzzling and struggling over. And so I would like to thank you all very much for the work you do, for the opportunity to come here and to talk to you. This is our group has been closed down now because we were funded by the Department of Energy, who developed imaging tools and who now have changed the mission. So this big group that we had here at Brookhaven, and it's really a multidisciplinary group. We have chemists and physicists, we have physicians, with psychiatrists, psychologists, engineers, all kinds of people, and a lot of students working together on this common problem. And during the time that we were supported both by the National Institutes of Health and the Department of Energy, and the Nora Volkoff's part, which is in the center there. Her group moved down to NIH, so they're trying to reconstitute this work, and they will reconstitute this work down in Bethesda. So I thank you all very, very much. And if you have questions, I'd be happy.
Shasta, do you want to command the question microphone? This one right here. Thank you very much. It was a great presentation. We have a lot of time for questions, so we'll start with people in the room, and then we'll go on to the people on remote.
Oh, we're on Long Island. Did I talk into the right place?
Yeah.
Okay. Yeah, it's one of the national laboratories. They support big science, big machines, big accelerators for understanding the early universe, that sort of thing. Yeah. But I also have supported. They supported atoms for peace and atoms for medicine and isotopes for medicine, which is how we got to develop radio tracers for imaging and for nuclear medicine. So if you have a brain scan or a bone scan, those kinds of things were developed at places like Brookhaven National Laboratory.
Thank you for the talk. Just a quick question. Have they found anything organic or natural that would stimulate more centers?
Yeah, that's a really
question.
Have they discovered any way of increasing and tuning up the dopamine system without actually stimulating craving and addiction? That's the big problem, is that these things stimulate craving. Because you think maybe we could just add a little bit, a molecule like methylphenidate or something. They seem to elicit craving. But I think what we need to do is take an approach that takes into account that you have different stages of addiction. You have the binging, and then the withdrawal, and then the craving, and then the cycle, that people may be in different cycles of this, and that you have to also work on the strengthening the frontal cortex, drugs that will help with strengthening there, and just kind of thinking of giving coping mechanisms to people when they're exposed to a drug. Okay, what do? You want to stay away from the drug. You think about it ahead of time, and I think that's things that psychologists and social workers can help people with is developing coping mechanisms. But we would very much like to be able to stimulate that dopamine system. There's evidence in animals that calorie restriction is helpful. Exercise is also helpful. So those kind of things that we always talk about as healthy lifestyle. That's a very good question.
The question is, I understand that chemicals change the brain. But the brain, from what I understand, brain chemistry is part of the loss of control. But would that loss of control also point to loss of free will?
That's a very good question, because the first time a person takes a drug, it's a choice, right? It's a conscious choice. But after a while, after the addiction sets in, people will tell you, I want to stop smoking, but I can't stop smoking, or I want to stop taking this drug, and it's cost me my job and my family. But it goes into this loop where they can no longer control it. So that's when I think it becomes a disease. Because we all know people, well, you can just think of drinking. There's an old number, 90 million social drinkers and 10 million alcoholics. About 10% will go on and become addicted. But there are people who can try a drug and experiment with a drug and then not become addicted. So there's this biology that goes along with it. But of course,
it's interesting, there's something called shippers, that people that just smoke a small amount of cigarettes and don't get addicted, although majority of people smoke a lot of cigarettes, and majority of people use cocaine to become addicted.
They do become addicted, and especially if they take it by, I think when it became a real problem with the crack and the crack smoking, because it got into the brain so rapidly, it was easy when didn't have to have needles. That's what's easy about cigarettes, it's as close as a match. And so you don't need anything complex.
We have a question from UTEP. Can you?
Yes, hi, can you hear us?
Yes.
Okay. You mentioned that nicotine can get into the fetal brain, and in the 9th month post-conception, there's dramatic changes in fetal brain development. Can you talk a little bit about what you might expect to be going on in the brain that last month of development when exposed to nicotine?
It's probably in there as the brain develops. This was in the 3rd trimester of these monkeys. One of the things you can speculate on is this thing that I touched on at the end, where this aromatase, where nicotine inhibits aromatase. So imagine a mother smoking, a pregnant woman smoking, and the inhibition of aromatase during this critical time when the male fetus is undergoing a transition to male behavior, for example.
But there's multiple developmental abnormalities of fetuses exposed to nicotine. And, I think you look at it historically, there were ads for pregnant women to smoke in the 60s. And, people thought it was relaxing and good for them. And how much have we learned so far, so many, few years?
And I think still 17% of pregnant women continue to smoke. Wow. Probably not because they want to. It's, again, this free will thing. Is they can't stop. It's painful.
Thank you.
Thank you.
Nice talk, but I have a question about monoxide.
Monoamine oxidase, right.
That's how specific it is. It's the other organs.
Oh, yes. Yeah, it inhibits, in fact, I don't have it in my slide deck, but if you do a whole body image of a smoker next to a non-smoker, it's the whole thing. Yeah. Oh, yes, it inhibits, but only partially. So we don't know the consequences of that. But it's very curious. It was very curious to find it accidentally. We didn't know what was going on. Yeah.
Is there any cross that can use the
yes, I think that's sort of similar to what the other gentleman asked. We don't know of anything. You can do it genetically. And exercise and calorie restriction has some evidence of being good for the dopamine system. So, yeah.
Oh, okay. Go ahead, Max.
There have been studies with PET imaging showing smoke cigarette nicotine deposit in the lung and then into the circulation. There was a device imaging showing deposit.
Right, yes, right, yes. I think a lot of it's going to be in the airways too, but I think enough get into the bloodstream to get into the brain and make that spike. But that's a very good question: is how rapidly do these aerosols they mimic actually smoke nicotine?
And
I don't,
Dr. Shao and I are looking at that actually.
Yeah, I think studying
Funding agencies will agree.
Yeah, I mean, one of the things was impressed me looking at Brody's study is that you can look at, I mean, there must be some nicotine coming out in that vapor. So it's secondhand smoke, the secondhand nicotine, not secondhand smoke, but secondhand nicotine, which I'm sure someone should be looking at that.
We are.
Good.
On the second, the first aspect of the cognitive disorder and the brain, what do you think about the new approach in clinical practice for the use of the drug replacement agents and the use of calcium ion to overcome the addiction?
Sorry, I didn't understand the use of what?
The use of drugs also replacement, like nicotine, for example, alcohol consumption or alcohol dependence in patients. How does that work? Because actually, the only pathway is the same. The check economic is the same on the brain. And isn't there the likelihood of the individual also being from the replacement drug?
Well, I think the replacement is like the replacement. It's a legal. It's like methadone. It's a replacement. It allows the person to function in society. It's not a cure because it's a replacement, but it's probably one of these harm reduction. It's better than he was being in the street doing the heroin. So, yeah, I know what you're getting at. It's not a cure, but it's something that's helpful if it's widely available.
Do you want to speculate about the high aromatase and lower aromatase and lower estrogen? You would expect maybe less breast cancer, but we don't see that. It's just because the other effects of the tobacco is just so detrimental in cancer.
I think so, but that would be something to look at. But I've talked with some epidemiologists, and they said there isn't an effect. So if
there's more breast cancer.
Yes. I don't know that there's more, but there certainly isn't less. And so, I think the effects of cigarette smoking overwhelm almost everything else that you can look at.
And it is the nicotine itself that inhibits aromatase or something else?
Isn't that interesting? I think it's really
so we had
something to think about.
We have some experiments looking at mice getting breast cancer cells implanted in them and giving them nicotine in a high-fat diet and combinations. And they seem to get a little less cancer growth in very small numbers of animals when they did get the nicotine. That might be an explanation of why if they might have lower estrogen levels.
Yeah, I don't know why this enzyme's been studied a lot in finches and fish, but not so much in humans.
A couple more questions. Anybody from remotely have a question? No? No? Any concluding words you want to mention, or
no, just
advice
keep,
to our colleagues
keep
here?
Keep up your work. Don't get discouraged, because this makes a huge impact on everything that we do, and on our ability to function as a society in a productive way.
Great. Well, thank you very much, Dr. Fowler. You actually did a great job with the
Wow.
Right, and then you got really good.
Yeah, I got good.
Yeah. Grab that off.
Thank you. Yeah, the key was getting rid of